Okay, so I'm going to get started. So my name is Angela Abitua, and I'm an outreach scientist at Addgene. And today I'm going to talk about the importance of material sharing, and I'll go through a few examples of how much of an impact material sharing makes to accelerating science. And then at the end, I'll talk about a few ways that you can make your own science more open and reproducible. So what do we mean when we say open science? So there are a lot of definitions out there that exist, and sometimes it can vary by the scientist who may be working in a different field from other scientists. But one of the key principles of open science is the idea of increasing access and usability of research. And to apply this idea towards the entire research process. So starting from the very beginning when you're developing your hypothesis and designing your experiments to the very last steps of publishing your paper. And another big takeaway from open science is the idea of sharing and collaboration, and to do this as early as possible because it really helps to speed up science. And so by practicing open science, there are many benefits. One of the biggest one is just improving the way that we do research. And this results in faster and more inclusive dissemination of knowledge to other scientists and to potentially to the general public. And it results in greater impact and reuse of research resources, as well as increased research reproducibility. And so this image here to the right is a modified graphic that I took from the Foster Open Science Learning Portal. It's a great resource. I recommend that you check it out if you haven't seen it before. And this graphic shows the many facets of open science. And so many of these components you may have thought of immediately when you thought of open science. So you might associate it with open science, such as open access articles, open data, open source. But one component that's missing from this, I think, is open research materials. And so this is a topic that isn't talked about a whole lot in the open science community, but I think that it should be, and it's something that we care a lot about at Addgene. And when I say open materials, or when I speak of materials, I mean biological materials like reagents that life scientists use in the lab, like plasmids, antibodies, cell lines. And so one example of how research can be slowed down when there's inaccess to materials or even protocols is from the Cancer Reproducibility Project. So this was a big project it was a collaboration, and the goal was to reproduce 50 high impact cancer papers in just 1 year. But this ended up dragging out to 5 years, and they eventually had to shrink down their study to just replicating 18 of these original high impact cancer papers. And they had to do this because they realized that it was just taking way too long to get the information and materials that they needed from those original authors. They spent way too much time trying to get those materials like cell lines and plasmids, or they wasted time having to remake the materials themselves. And so simply not having proper access to materials and protocols can really slow down research. And when it comes to the sharing of materials, this traditional way of sharing reagents has many barriers. So, we have the corresponding author that publishes their research, and they make their reagents available upon request. Scientist reads the publication and requests the material, And sometimes this can take a very long time to happen for them to receive those materials. So, on the receiving end, this scientist can wait weeks or months to receive the material from the author, or they have to spend time to recreate it themselves. And for the author, there's a lot of responsibilities. They have to track and store all the materials. They have to authenticate the materials to make sure that they're shipping the correct reagent to the requesting scientist, and they have to deal with legally distributing and shipping this material to other scientists. And if this material is very popular, then that can be a big burden on the author who is publishing the paper. And so repositories are a part of the solution. For the author, if they deposit their materials in a repository, the repository handles all of those logistics that I just mentioned, so that the author no longer has to worry about those things, and they can focus back on their research. And for the requesting scientists, they're able to receive the materials, the correct materials, more quickly. And so this is exactly what Addgene does. We help scientists share their materials, their research materials and associated data with other researchers. And for those of you who are not familiar with what we do, Addgene is a nonprofit, and our mission is to accelerate research and discovery by improving access to useful research materials and information. And so in terms of what materials we offer, we have a large collection of plasmids, which are circular pieces of DNA used for research. And since 2004, we've collected deposits from many scientists from around the world, and our collection is now over 80,000 plasmids, which also includes pooled screening libraries, plasmid kits, and we also have a ready-to-use AAV, adeno-associated virus, and lentivirus preps, this prep service for scientists, which the AAV has been very popular amongst the neuroscience community. And we also have several educational resources on our website, including curated guides, written and video protocols. The video protocols can be viewed on YouTube. We have many blog posts and ebooks. And so now I'm going to jump into some examples of how much of an impact material sharing can make in research and how it can speed up the process. And so here's a really great example of how Addgene has played such a big role in enabling the rapid adoption of a technology like CRISPR-Cas9 by helping scientists to quickly share and gain access to these plasmids. And so to date, scientists have deposited over 10,000 CRISPR plasmids from over 900 labs in 29 countries around the world. And we've distributed over 180,000 CRISPR reagents to 87 countries, to labs in 87 countries. And if you're interested in learning more about this, you can read this article that was published in the CRISPR journal. And I also wanted to add that of these CRISPR-based tools that are available through Addgene, there were 2 labs that developed CRISPR-based diagnostics. And one was for detecting DNA, known as DETECTR, which was developed in Jennifer Doudna's lab at UC Berkeley. And then one for detecting RNA, known as SHERLOCK, that was developed in Feng Zhang's lab at the Broad Institute. And if you're interested in learning more about those diagnostics, you can read about it in our blog. And so these CRISPR-based diagnostics that had plasmids that were available through Addgene, it turns out that a few weeks into this COVID-19 pandemic, as it was spreading in the US, it turns out that these two diagnostic tools were being developed by those original labs to detect COVID-19. And because these plasmid tools were also available through Addgene, they were also being adapted by other labs in other parts of the world to make low-cost and quick detection assays for COVID-19. And since the beginning of the pandemic, despite the reduced deposits and shipping of plasmids due to lab closures around the world, Addgene has received over 2,000 plasmids for COVID-19 research and over 5,000 requests from about 50 countries. And if you're interested in learning more about this, you can read this article which is also published in the CRISPR journal. And so we've also created a COVID-19 resource page, which you can visit here at this link. And this page is updated daily, and it also includes, it makes it easier for researchers who are studying coronavirus to quickly find the plasmids that they need. And there's also protocols and additional resource links. And some of these plasmids have also been opened up to industry, which means that we can make these materials more openly and widely used. And so, as I mentioned, this is something that Addgene has been working hard to work with COVID-19 researchers to distribute these useful plasmids for coronavirus-related research. And so, since March, over 1,000 new plasmids related to COVID-19 have been made available for request. And of those new plasmids that have been deposited, 90% of those plasmids are available to industry-based scientists. And again, this is making it open to many more scientists than we could by only offering it to academic or nonprofit organizations. And so if you're interested in learning more about the industry plasmids or the plasmids available to industry, you can visit this website. And then I also want to point out that in addition to the COVID-19 related plasmids, we also have a growing number of collections that are available to industry, such as the Michael J. Fox Foundation collection for studying Parkinson's research, the Structural Genomics Consortium, and the Resolute Consortium collection, as well as many fluorescent protein plasmids that are available through Florida State University. And, all of this, the opening of these plasmids to industry wouldn't be possible without the academic institutions making their plasmids available to both for-profit and nonprofit use. And so there's a growing number of academic institutions that are starting to share these materials and make them more open to all scientists, including for-profit scientists, for research purposes. And so if you're interested in learning more about the industry plasmids, I encourage you to read this blog post, this recent blog post on the Addgene blog. And so I'm going to talk about one other open science related project that we've been working on at Addgene. So if you published a paper in the past few years, you may have noticed that the publisher may require you to deposit your data. And so at Addgene, we've been working with publishers to update the material sharing policies to recommend the deposition of materials with centralized repositories like Addgene. So currently there are over 50 journals that recommend depositing these materials to these repositories. And if you're interested in learning more about this, you can read this article with this by clicking or by typing in this DOI link. to learn more about how publishers can really make a big difference in increasing the, or making material sharing better. And so this is just, one additional way to encourage scientists to practice more open and reproducible science, because it's coming from the publishers. And so now I'm going to go through a few ways that you can make your own research more open and reproducible. So the first one is to deposit your materials if you work with biological materials. So it's generally free to deposit. At Addgene, it's definitely free to deposit. As I mentioned before, like this, ensures that the materials you have are safely stored somewhere. And, we take care of the shipping and distribution of your materials. You also get to keep track of who receives all of those materials. We take care of the tech transfer. And we always encourage you to deposit before publication. Because it can make the manuscript writing process easier. And it also results in increased citations. And another way that you can make your science more open is, of course, to deposit your data. And so here are just a few general purpose repositories that are out there with different features. And if you're looking for something more specific, you can also go to the Registry of Research Data Repositories, re3data.org. So that's a great resource to find something that's more specific to your data set. And I also wanted to point out that Addgene also has a data repository of its own. So in 2019, we launched the AAV Data Hub. And so AAV are, which stands for adeno-associated virus. They are very useful tools that are used a lot by neuroscientists for expression studies and manipulation of neurons. And many scientists have benefited from Addgene's ready-to-use AAV viral prep service. And so we launched this data hub as basically an open platform for researchers who are using AAV to share their experimental data with other researchers. And we also accept negative data as long as the proper controls are included. And this is a way for us to help researchers find the right AAV tools for their experiments and to help them design their experiments. And so if you're interested in learning more about the AAV Data Hub, you can visit datahub.org or datahub.addgene.org/AAV. And another way that you can make your science more open, as I mentioned earlier, one of the difficulties with the Cancer Reproducibility Project is they simply weren't able to get a hold of some of these protocols for their studies. And so as with data and materials or reagents, you should really consider using a repository to deposit and share your protocols. And so here are just a few examples of some repositories that you can use. So there's Protocol Exchange and Protocols.io. I know a little bit more about Protocols.io, and there it's a really nice repository that allows you not only to store your protocols, but to easily share it with others. Because every time you publish a protocol, you get a DOI, which is a permanent link that people can also cite. And then it also takes care of versioning, and it makes it easier for you to share those protocols with other scientists. And at Addgene, we have over 30 protocols available on our website that cover, range from techniques from, basic techniques like pouring a gel to more advanced techniques like how to prepare a library. And these are also, all of these protocols are also shared on protocols.io. So if you're interested, you can check out the link right here to view all of those protocols. And finally, another thing that you can do to make your science more open is to learn more about open science. So here are a few other resources for, they're great resources for learning more about open science, and as well as reproducibility. So I hope that after this talk, you've gained a better understanding of the importance of material sharing and the impact that it can make, and that you've gained some inspiration on ways that you can make your own science more open and reproducible. Thanks everyone for joining the webinar today. And I hope that this was helpful, and this is recorded, so we'll be sharing this with you afterwards. Thanks everyone, bye.